Welcome back friends. Welcome back again to one more of the lecture session from this channel. So in this lecture session we will be studying regarding genetically modified crops from biotechnology and its applications in detail. So here, what are genetically modified organisms? The specific living organisms whose genes have been altered by manipulation. Such specific living organisms, they are termed as genetically modified organisms. And here, some of the genetically modified crops, they may include Bt cotton, Bt tomato, Bt brinjal, potato and soybean. So here all these are the various genetically modified crops. Is it clear? So let me write it on the board. Specific living organisms. Specific living organisms whose genes have been altered by manipulation such specific living organisms are termed as genetically modified organisms. Genetically modified organisms. Genetically modified organisms, right? And here, some of the genetically modified crops, they may include Bt cotton. Some of the genetically modified crops may include Bt cotton. Is it clear? Bt cotton, Bt tomato, Bt brinjal, potato and soybean. Bt brinjal, potato and Bt tomato and soybean. So here all these are the various genetically modified crops. So at first we will be studying regarding Bt cotton. So here what is Bt cotton? So let me write it here. Bt cotton. So here Bt cotton, it is one of the genetically modified crop which has successfully designed by taking the advantage of a specific microbe termed as Bacillus thuringiensis. Is it clear? Bt cotton, it has successfully designed by taking the advantage of a specific microbe termed as Bacillus thuringiensis. So here Bacillus thuringiensis, it is a specific microbe which has a specific endotoxin gene termed as Bt toxin. So here when the specific Bt toxin, once when it undergoes successful expression, it is capable to regulate the expression of a specific protein termed as insecticidal protein. So here the specific insecticidal protein, most commonly it targets many number of targeted insects like lepidopterans, coleopterans and even diopterans. Is it clear? Bacillus thuringiensis contains a specific endotoxin gene termed as Bt toxin. So here, once when the specific Bt toxin, once when it undergoes successful expression, it is capable to regulate the production of a specific protein termed as insecticidal protein. So here the specific insecticidal protein most commonly targets many number of the specific insects like lepidopterans, coleopterans and even the diopterans. The specific insecticidal protein targets many number of targeted insects like lepidopterans, coleopterans, especially like beetles, and the other one is diopterans, especially like flies and mosquitoes. Here, a question arises in your mind. Why the specific endotoxin gene, which is a part of Bacillus thuringiensis, it won't have any negative effect within the Bacillus thuringiensis itself? Because initially, here we need to extract the endotoxin gene from the Bacillus thuringiensis. After extracting the endotoxin gene from the Bacillus thuringiensis, now here we need to transfer the endotoxin gene into the specific host plant. So here, once when the endotoxin gene, once when it has successfully transferred into the specific host plant, now likewise the other genes of the host plant, even the foreign desirable gene, which is nothing but the endotoxin gene, it undergoes successful expression. And here, the specific targeted insects like lepidopterans, coleopterans and diopterans, once when they target the leaves of the host plant, the endotoxin gene initially which is under inactive form within the Bacillus thuringiensis, it undergoes successful expression within the intestinal gut of these targeted insects, because within the intestinal gut of these targeted insects alkaline pH is present. So in the presence of alkaline pH, here the specific endotoxin gene initially which is under inactive form, now here it remains in active form. So here once when the endotoxin gene, once when it remains in active form within the alkaline pH of the intestinal gut, now here the endotoxin gene, it is capable to regulate the expression of a specific protein termed as insecticidal protein. So here these insecticidal protein most commonly they tag on the surface of the epithelial cells of the intestinal gut. So here once when the specific proteins, once when they target the epithelial surface of the intestinal gut, now here these specific proteins, they are capable to cause specific pores. So here in the presence of the specific pores, here we can observe swelling of the intestinal gut. So here once when the swelling of the intestinal gut takes place, it ultimately causes the death of the living organism. Is it clear? So let me write it on the board. Endotoxin gene which is a part of Bacillus thuringiensis remains in inactive form within the Bacillus thuringiensis. Right? But here, once when the endotoxin gene, but here once when the endotoxin gene when it successfully transfers, when it successfully transfers into the intestinal gut of the targeted insects, transfers into the intestinal gut of targeted insects, it remains in active form. It remains in active form. Is it clear? So let me erase the board here. And as already mentioned, in the presence of alkaline pH within the intestinal gut of the targeted insects, now here the endotoxin gene, which is under inactive form initially within the Bacillus thuringiensis, now it remains in active form. And here the specific endotoxin gene, it is capable to cause specific pores on the epithelial surface of the intestinal gut. So here, because of the presence of pores on the epithelial surface of the intestinal gut, it is capable to cause swelling, and even ultimately it leads to the death of the targeted insect. Is it clear? In the presence of alkaline pH, the endotoxin gene remains in active form, and capable to cause specific pores on the epithelial surface of intestinal gut. And here, because of the presence of pores on the epithelial surface of the intestinal gut, it causes swelling, and here ultimately it causes the death of the targeted insect. Epithelial surface, it causes swelling, and ultimately it leads to the death of the targeted insect. Right? So here there are many number of endotoxin genes which are a part of Bacillus thuringiensis, and here all the endotoxin genes which are a part of Bacillus thuringiensis, most commonly they are host specific. According to the specific host plant, here the endotoxin gene which is a part of Bacillus thuringiensis, it is successfully isolated and it is transferred into the specific host plant. Is it clear? There are many number of endotoxin genes which are a part bacillus thuringiensis, which are a part of bacillus thuringiensis. And all the genes which are a part of bacillus thuringiensis, are host specific. So here, according to the desirable host plant, here the endotoxin gene, it is successfully isolated and it is transferred to the specific host plant. And here most commonly, all the endotoxin genes which are a part of the specific bacillus thuringiensis, they are encoded by a specific gene termed as cry genes. Is it clear? Most commonly, the endotoxin genes which are a part of bacillus thuringiensis are encoded by a specific gene termed as cry gene. So here, some of the cry genes which are very specific to the concerned host plant, they may include cry1AC and cry2AB. So here cry1AC and cry2AB, these are the specific endotoxin genes which are very specific to cotton bollworm. Is it clear? So let me write it here. Some of the endotoxin genes may include cry1AC and cry2AB, which are very specific to cotton bollworm. Right? So here likewise cry1AC and cry2AB, even the specific endotoxin gene like cry1AB, it is a specific endotoxin gene which is very specific to corn borer. Is it clear? Likewise cry1AC and cry2AB, even cry1AB is very specific to corn borer. Is it clear? So here this is regarding genetically modified crops, and then after here we will be studying regarding pest resistant plants. So here what are pest resistant plants? So let me arrange the things here. After here we will be studying regarding pest resistant plants. So here what are pest resistant plants? So here there are many number of nematodes which are capable to target many of the living organisms like plants, animals and even the humans. So here one of the specific nematode which most commonly targets the specific tobacco plant, here it is nothing but Melatogne incognita. So here the specific nematode most commonly targets the root of the tobacco plant. So here when the specific nematode, when it targets the root of the tobacco plant, it is capable to reduce the crop yield. So here in order to overcome the problem, here there is an availability of a novel strategy termed as RNA interference. So here by taking the advantage of RNA interference, here we can overcome the problem. Is it clear? So let me write it on the board. There are several nematodes which are capable to infect many of the living organisms like plants, animals, and even humans. So here one of the specific nematode which most commonly targets the root of the tobacco plant, here it is nothing but Melatogne incognita. One of the specific nematode which targets the root of the dicotyledonous plant, which is nothing but the tobacco plant. It reduces the crop yield. So here, in order to overcome the problem, there is an availability of a novel strategy termed as RNA interference. So here RNA interference, it is a type of cellular defense mechanism which is most commonly observed nearly in case of all the eukaryotes. In order to overcome the problem, a specific novel strategy termed as RNA interference do exist. So here RNA interference is a type of cellular defense mechanism which is observed nearly in case of all the eukaryotes. Which is observed nearly in case of all the eukaryotes. So here, once when the specific nematode, when it undergoes successful expression within the host plant, it is capable to regulate the expression of mRNA. Is it clear? So let me show it on the board. So here, once when the specific gene, when it undergoes successful expression within the host plant, it is capable to regulate the expression of mRNA. Right? Now here, once when the specific mRNA which has successfully synthesized during the transcription mechanism, once when it participates in the translation mechanism, it is capable to regulate the production of the specific protein. So here, once when the specific protein, once when it has successfully synthesized within the cytoplasm, it indirectly suggests or it indirectly says that the disease causing gene, it has successfully expressed within the host plant. Now here, we need to inhibit the expression of the protein within the host plant by taking the advantage of a specific novel strategy termed as RNA interference. So here RNA interference most commonly targets the inhibition of the protein formation. Is it clear? RNA interference most commonly inhibits the gene expression by inhibiting the translation mechanism. By inhibiting the translation mechanism. So here, in order to inhibit the translation mechanism during the protein synthesis, now here there is a requirement of a specific vector termed as agrobacterium vector. So here, by taking the advantage of this specific vector, a specific gene, it is successfully inserted here. So here, once when the specific desirable gene or once when the specific desirable nematode gene, once when it has successfully inserted into the agrobacterium vector, now here the specific agrobacterium, likewise the other bacteria, even it is also capable to infect the tobacco plant. So here within the agrobacterium vector, here there is an availability of a specific nematode gene. So here once when the specific nematode gene, once when it undergoes expression, it is capable to regulate the expression of a specific RNA termed as miRNA or microRNA. Is it clear? So let me write it here. Agrobacterium vectors are most commonly used to transfer the desirable nematode gene within the host plant. So here, once when the specific gene, once when it undergoes expression within the specific host plant, now here it is capable to regulate the expression of a specific RNA termed as microRNA or miRNA. So here the specific microRNA or miRNA, it remains quite complementary to the specific mRNA. Is it clear? So let me show it here. So here it is nothing but what? miRNA or microRNA. So here once when the specific microRNA, which remains quite complementary to the specific mRNA, once when it is it binds to the specific mRNA, now here the ribosomal subunit, it is unable to bind to the specific mRNA. Once when the miRNA, once when it remains quite complementary to the mRNA, now here there is an availability of double stranded RNA. In the presence of double stranded RNA, the ribosomal subunit, it is unable to bind to the specific mRNA. So here once when the ribosomal subunit, once when it is unable to bind to the specific mRNA, now here there is no chance for the formation of a specific protein during the translation mechanism. Is it clear? So let me arrange the things here. Now here, once when the specific microRNA or miRNA, once when it successfully binds to the specific native mRNA, now here the ribosomal subunit, it is unable to bind to the specific mRNA. So once when the ribosomal subunit, once when it is unable to bind to the specific mRNA, now here there is no chance for the expression of a specific protein. Is it clear? Once when the microRNA binds complementarity to the specific mRNA, now the ribosomal subunit, is unable to regulate the synthesis of protein. unable to regulate the synthesis of protein during the translation mechanism. Synthesis of protein during translation mechanism. Translation mechanism. So here, one of the consequences of this particular novel strategy, here it is nothing but the specific parasite, which is nothing but the nematode, it is unable to survive within the transgenic host in the presence of the specific interfering RNA. In the presence of the interfering RNA, the specific nematode, it is unable to survive within the transgenic host. So in this way, the RNA interference mechanism, it is capable to overcome the negative effect of many of the disease-causing pathogens in case of the host plants. Is it clear? So let me write it here. One of the consequence of this novel strategy is nothing but the specific parasite is unable to survive within the transgenic host in the presence of the interfering RNA. Is it clear? So here this is regarding genetically modified crops and pest resistant plants from biotechnology and its applications in detail. And in the next upcoming lecture session, we will be studying regarding role of biotechnology in medicine in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.